Good morning, everyone. My name is Marc Applehans. I'm a postdoc fellow here, and I just started 4 months ago. So I want to summarize the work of my first 4 months here in the botany department. I started in February, and before that, I did my master's in Germany in Marburg. Then did my PhD in Leiden in the Netherlands, and I just graduated in November last year. So what my supervisors and I are interested in is Pacific and especially Hawaiian biogeography, and we aim to understand and explain the current distribution of plants, and that involves questions such as what's the geographical origin of plant lineages like species, genera, families, what's the age of these lineages. Can everyone understand me? Yeah. A bit louder? Okay. And the next question would be how often did a particular plant lineage colonize islands or an island group? How did they get there? Was it a single long-distance dispersal event? Were there islands available in short distances that could serve as a stepping stone? Or was it a vicariance event? Vicariance means that there was a physiological barrier emerging, like a new mountain range or tectonic plates breaking off, so there was no genetic exchange anymore, which eventually leads to speciation. One question that is particularly interesting for Hawaii because of its very isolated position is Hawaii a dead end for dispersal? And a very powerful tool to answer all these questions are molecular phylogenetic trees. I was told to explain what that is because not all of us are biologists here. I think this picture is quite self-explanatory, so it's a diagram that explains evolutionary relationships between different organisms. I put the word dated in brackets before, so the method of molecular dating explains, or if you have a fossil of, in this case, the most recent common ancestor of reptiles and birds, you can infer from that, if you have a very well dated fossil, you can estimate all the other branching patterns in this phylogenetic tree, and therefore put ages on lineages where you don't have a fossil of. Okay, so that was the research questions. Why do we study the genus Melicope? In this list, you see the largest genera of the Hawaiian Islands, Melicope on number 4, but it's as more or less as speciose as number 2 and 3. So it's a very important element of the Hawaiian flora. If we look at the whole distribution range of the genus, we see it's common in many Pacific islands, as well as in Australasia, Southeast Asia, and interestingly also a couple of species in Madagascar. It's quite species rich. In total there are 230 species, so it's the largest genus of the citrus family. And I mentioned already the number of species in Hawaii. Another center of distribution is New Guinea, with almost 60 species and 44 of them only occurring there. And so summing up, it has a very large distribution range. It's quite speciose, so it's very well suited to answer questions on biogeography in the Pacific area. I have just one slide of my results so far, so it's only been 4 months since I'm here. And I want to point out only 2 results. So you see what I'm showing here is a phylogenetic tree based on DNA sequence data of both the nucleus and the chloroplast combined. You see these species in the blue box, and those are the ones from Madagascar, so the ones that are outside of the normal distribution range of the genus. They clearly fit into Melicope, and they are quite young, so our age estimates are between 2.5 and 6 million years. So this definitely doesn't have anything to do with any tectonic movement, for example, the drifting of the Indian plate or something. It's much, much too young for that, so it is a fairly recent long-distance dispersal event. You see these species in the red box. Those are the ones from Hawaii, and they all originated from one common ancestor, and there was probably one colonization event to the Hawaiian Islands. But nested within this red box, you see these 2 yellow boxes, and those are species from the Marquesas Islands in the southern Pacific. And as I mentioned before, Hawaii was often regarded as a dead end for dispersal, but in this case, it's clearly not the case. So we have 2 independent dispersal events from Hawaii to the Marquesas Islands. Interestingly, the Hawaiian lineage is quite old. The estimates are 6 to 11 million years. And the current islands in the Hawaiian archipelago are not older than 5 million years. So Kauai and Niihau are the oldest with about 5 million. But, well, I think you know that the Hawaiian range, or the Hawaiian, the Hawaiian range is older than that, and there were islands before, and islands such as Necker and Nihoa were present 5 to 10 million years ago, and they might have been the source of the Hawaiian Melicope. Okay, what's next? So I'm here for at least 1 year, and so more than half of it is still to do. So we want to include more species from outside Hawaii to broaden the biogeographic picture. And for that, we, of course, contacted colleagues, but we also plan a field trip to Papua New Guinea later this year. And another thing we want to find out is, was the colonization in the Hawaiian Islands from the oldest to the youngest islands? That's something we often observe in many plant and animal lineages, and we have this picture in our plants as well, but we need a bit more sequence data to really be certain about it, so we need a bit more sequencing to answer that question. Okay, and with that, I'd like to thank you all for coming and for listening. Thank you. The question was, how did I calibrate the molecular clock? Well, I indicated in one slide, I didn't say it, that it's fossil calibrated. So we have fossils not from Melicope, but from a related genus. And so we used that fossil as an age calibration. But also we used age estimates from an earlier study to calibrate or to set the base of the phylogeny. So the root age was fixed.